Welcome to the Biology Domination Podcast by Hidden Wisdom Academy Biology, your audio companion for the NEET 2026 crash course. I'm your host, and today is episode 13, covering one of the most high-weightage chapters in the entire NEET syllabus, photosynthesis in higher plants. This episode is designed so you can study even when you're not at your desk, whether you're commuting, eating, or taking a break. Pop in your earphones and let's get this chapter done. Today we'll cover everything systematically. Pigments, the complete Z-scheme light reactions, the Calvin cycle, C4 and CAM pathways, photorespiration, and factors affecting photosynthesis. Every single concept that NEET can and will test. Let's get into it. Let's start with photosynthetic pigments, because NEET loves asking about them, and they're conceptually connected to everything else in this chapter. Inside the chloroplast, the thylakoid membranes contain 4 key pigments. Chlorophyll A, the primary pigment, it absorbs light most strongly at 430 nanometers in the blue region and 662 nanometers in the red region. Critically, chlorophyll A is the only pigment that directly performs the photochemical act, the actual energy conversion step. Then we have the accessory pigments, chlorophyll B, which absorbs at 453 and 642 nanometers, xanthophylls, which are yellow, and carotenoids, which are yellow-orange and also serve a protective function by absorbing excess light that could otherwise damage the cell. These accessory pigments absorb light at wavelengths that chlorophyll A can't, broadening the range of usable sunlight. They then transfer that energy to chlorophyll A. Now, paper chromatography. This is the technique used to separate these pigments. The pigments travel up a chromatography paper at different rates based on their solubility. Carotenoids are the most soluble and travel the farthest, highest RF value. Chlorophyll B is the least soluble and stays lowest RF value. The action spectrum shows the rate of photosynthesis at different wavelengths. The absorption spectrum shows which wavelengths each pigment absorbs. The fact that these two spectra nearly overlap for chlorophyll A is direct experimental proof that it is the primary photosynthetic pigment. This is a classic NEET conceptual question. Now let's talk about the Calvin cycle, where the actual sugar making happens. These are the dark reactions, but that name is misleading. They don't need darkness, just don't directly need light. They happen in the stroma of the chloroplast. The cycle has 3 stages. First, carbon fixation. The enzyme Rubisco catalyzes the combination of one CO2 molecule with RUBP, a 5-carbon sugar, to produce an unstable 6-carbon compound that immediately splits into 2 molecules of 3-PGA, 3-phosphoglyceric acid. First, stable product. Please tattoo this on your brain. Second, reduction. These 3-PGA molecules are reduced to G3P, glyceraldehyde 3-phosphate, using ATP and NADPH from the light reactions. Some G3P goes on to form glucose. The rest is used for regeneration. Third, regeneration of RUBP. The remaining G3P molecules, using more ATP, are converted back into RUBP to keep the cycle going. For every 6 CO2 fixed, producing 1 glucose, the cycle uses 18 ATP and 12 NADPH, and Rubisco is literally the most abundant enzyme on Earth. Now, the C4 pathway. C4 plants solve the photorespiration problem using 2 brilliant strategies, a structural one and a biochemical one. Structurally, Cran's anatomy. The vascular bundle is surrounded by bundle sheath cells with large chloroplasts, which are surrounded by mesophyll cells. This physical separation of CO2 fixation stages is key. Biochemically, the first enzyme is PEP carboxylase, not Rubisco. PEP carboxylase fixes CO2 onto PEP in the mesophyll to form OAA, a 4-carbon compound. OAA is then converted to malate and transported to the bundle sheath cells, where CO2 is released and enters the Calvin cycle via Rubisco in a concentrated high CO2 environment. The result, Rubisco never encounters high O2 concentrations. Photorespiration is essentially eliminated. Let's go deep on photorespiration, the chapter's most misunderstood concept. Rubisco, as we said, is a dual-function enzyme. Its carboxylase function fixes CO2 into the Kelvin cycle. But its oxygenase function is its dark side. When O2 concentrations are high and CO2 is low, which happens on hot, bright days when stomata partially close, rubisco binds O2 instead of CO2. This produces a 2-carbon compound called phosphoglycolate. This compound is metabolically useless for photosynthesis. It gets shipped from the chloroplast to the peroxisome, where it's partially processed, and then to the mitochondria. In this journey, CO2 is released. The plant breathes out what it just tried to fix, with absolutely no ATP or sugar produced in return. Photorespiration can waste up to 25% of fixed carbon in C3 plants under hot conditions. This is a massive evolutionary disadvantage. Now, CAM plants, crassulation acid metabolism. These are your desert plants. Cacti, pineapple, agave, bryophyllum. CAM plants face a dilemma. Open stomata for CO2, but lose precious water in the heat. Their solution is elegant. They open their stomata only at night, when temperatures are cool and water loss is minimal. At night, CO2 enters and is fixed by PEP carboxylase into OAA, which is then converted to malic acid and stored in the vacuoles. During the day, stomata are closed. No water loss, no photorespiration. The stored malic acid is decarboxylated, releasing CO2 directly into the Kelvin cycle. Same biochemistry as C4, but separated by time instead of space. C4 separates the pathways in different cells. CAM separates them in different times of day. This is a frequent NEET comparison. C4 is spatial separation, CAM is temporal separation. Let's cover the factors affecting photosynthesis and then end with the 7 most dangerous NEET traps from this chapter. Blackman's law of limiting factors states that the rate of photosynthesis at any given moment is limited by the factor present in the smallest relative amount. This seems simple, but NEET tests your ability to apply it in graph-based questions. Light intensity. At low light, rate increases linearly. At the light saturation point, another factor becomes limiting and rate plateaus. Too much light causes photoinhibition. CO2 concentration, the most commonly limiting factor in nature. Atmospheric CO2 is only 0.03%. Small increases dramatically boost photosynthesis up to about 0.05%. Temperature. Optimal is 25 to 35 degrees Celsius. Above 40, enzymes begin to denature and stomata close. Water. Both direct and indirect effects. Directly, it's the substrate for photolysis. Indirectly, water stress triggers stomatal closure. Now, the 7 traps that have cost students marks in NEET. 1. First stable product in C3 is 3-PGA. Not glucose, not CO2, 3-PGA. 2. First stable product in C4 is OAA. 3. Primary CO2 acceptor in C4 is PEP, not RUBP. 4. Cyclic photophosphorylation produces only ATP, no NADPH, no oxygen. 5. Photorespiration occurs in 3 organelles, chloroplast, peroxisome, and mitochondria. 6. Only chlorophyll A performs the actual photochemical act. All others are accessory pigments. 7. PS2 is functionally first, despite being named second. Don't let the numbering confuse you. If you can answer a question on each of these 7 points, you will not lose a single mark from this chapter. And that brings episode 13 to a close. Today we covered every major concept from photosynthesis in higher plants that NEET 2026 can test. From the 4 photosynthetic pigments separated by chromatography, through the full Z-scheme light reactions with ATP synthesis by chemiosmosis, through the Kelvin cycle with rubisco and 3-PGA, through C4 Cran's anatomy and PEP carboxylase, through CAM's brilliant nighttime CO2 fixation strategy, through the wasteful disaster of photorespiration, and finally, Blackman's law and all the key limiting factors. If you listened to this entire episode, you have covered this chapter more thoroughly than most students will in their entire preparation. Seriously, take a moment to appreciate that. Episode 14 is respiration in plants, glycolysis, the Krebs cycle, the electron transport system, and fermentation. another heavy weightage topic with a lot of NEET traps. Share this episode with your study group. Subscribe so you don't miss a single day. And remember, 30 days of consistent effort will change your NEET result. You're already 13 days in. Keep going.